Okay, let's continue on muscle. And this should, this pretty much should finish the muscle chapter. Okay, so we looked at cardiac muscle. And again, I'm just going to talk a little bit about cardiac muscle. Because as I mentioned earlier, that we're going to have a whole, next semester, you'll have a whole chapter on the heart. Okay, so it's obviously found only in the heart, connected electrically by gap junctions. So skeletal muscle, remember skeletal muscle cells are electrically isolated from each other. Okay, they can be right next to each other and firing and they don't influence each other. But now with the heart, it has gap junctions, essentially electrical synapses, where ions can flow directly from the sarcoplasm of one heart muscle cell to sarcoplasm of another one. So it can excite its neighboring cell to threshold. That means that the muscle can contract in waves back and forth, which the heart has to do. Okay, it's involuntary, it's autorhythmic. The conductive cells fire all on their own. They got leaky sodium channels and they reach threshold all by themselves. And the fastest ones that do that are in the SA node, and that's the pacemaker for your heart. And almost exclusively aerobic metabolism. We talked about glycolytic fibers in skeletal muscle that really work anaerobically, but your heart has almost no anaerobic capability. It's very, very aerobic, very sensitive to oxygen concentration. Okay, and there it's also quite different in the sense that skeletal muscle cells are long cylinders. Heart muscle cells are branched, and skeletal muscle cells are multinucleated. Heart muscle cells have a single nucleus, and they have these intercalated discs where we have desmosomes and gap junctions. Desmosomes are like spot wells that hold the heart muscles together when they're contracting, and the gap junctions, which again allow electrical communication directly between cells. And like I said, they have lots and lots of mitochondria because they're very aerobic. Okay, with a smooth muscle. Fusiform shaped, the myocytes are fusiform shaped, that is like, how do you call it, fusiform? Thick in the middle and tapers at the end. They're also uninuclear. There's no sarcomers, Z discs, T tubules, very little SR. Actually, I should have mentioned in heart muscle cell also, very little SR. Skeletal muscles carry a calcium supply along with them in their SR, and so they're not influenced by external calcium. Heart muscle is very influenced by external calcium. Most of the calcium that turns on heart muscle comes in from the outside. And that's true for smooth muscle also. Very little sarcoplasmic reticulum. Most of the calcium, and calcium is a trigger for smooth muscle contraction also, as well as cardiac. It binds to troponin in cardiac and skeletal. It binds to a different protein that I mentioned a little bit later on in smooth muscle. Okay, capable of both hypertrophy, going larger, but also of hyperplasia, mitosis. Heart muscle cells and skeletal muscle cells are amitotic. They don't go through mitosis anymore. Once you're an adult, that is. But smooth muscle cells do. You can build more smooth muscle. And this is when you start doing aerobic exercise and you start building blood vessels to your muscles. Those blood vessels are surrounded by smooth muscle cells, and so you are making new smooth muscle. There's two types of smooth muscle, what's called multi-unit. It's the rarer form found in piloerector muscles, which is the muscles that cause goose flesh, which is completely useless. In the iris, large arteries and pulmonary air passages. And they act like skeletal muscle. Not that they're voluntary, but they have motor units that act independent of other motor units. And every muscle cell is innervated by a branch of a motor neuron. That's not true for what's called single unit smooth muscle, which is the more common type. And what happened there? Why are we? I think I stopped. Oh, here it is. Okay. No, where am I? Something's going on here. Hang on a second. Am I still screen sharing? I think I stopped screen sharing for some reason. Okay, here we go. Okay, so there's a form of what's called single unit smooth muscle. And that is, there's this motor axon with these verticosities coming by. And did we skip a slide? Hang on a second. No. Where these verticosities just like leak out neurotransmitter. There's no real synapses. It just leaks out and acts more like a paracrine than a neurotransmitter. And here's the microscopic arrangement. We do have actin, thin and thick filaments with myosin and actin, and they do overlap. But there's no, I said there's no Z lines, there's no A or I bands or H bands. And the actin filaments are attached to these so-called dense bodies. When a smooth muscle contracts, the filaments slide by each other and pull the dense bodies closer together. And they do so in a spiral-like motion. Okay, so a single unit, as you just saw a figure of, more widespread, found in most blood vessels, digestive, respiratory, urinary, reproductive tracts. Each cell electrically connected to each other via gap junctions, and the whole muscle acts as one. And this motor neuron comes by with these verticosities, and this neurotransmitter just leaks out, binds to receptors, causes contraction, and then the contraction of this cell has gap junctions in the next cell, like in cardiac muscle, and causes the next cell over to contract. So the whole muscle acts as one. That's why it's called single unit. Okay, stimulation, involuntary, and they can be stimulated by the leaking out of that neurotransmitter that comes by, but they can actually activate it also by hormones, and by chemical conditions, the amount of CO2, the pH, the amount of O2, so the stretch, and it can be innervated by branches of the autonomic nervous system. So as I said, no synapses. These verticosities and diffuse junctions, and these caveolae, which are like little pits in the cell membrane where we have neurotransmitter receptors. And so neurotransmitters, as I said, leak out of these verticosities, diffuse down to these to receptors in the caveolae, and that initiates a muscle contraction. So there's multi-unit on the left. You see this autonomic motor neuron, and it's innervating every single muscle cell with synapses. And then single unit on the right, we have these verticosities, and an autonomic motor neuron has these verticosities, and it just travels by, and there's these caveolae where, as I just mentioned, the neurotransmitter receptors are, and then each cell is linked by gap junctions. So the one next to the motor neuron gets stimulated, and then it stimulates the next one over, by gap junctions. Okay, contraction relaxation. Calcium is a trigger, but it comes from the extracellular fluid and binds to calmodulin in the cell, not troponin. So that's one big difference. Calmodulin activates something called myosin light chain kinase, which activates the myosin ATPase, which breaks ATP. Myosin binds to actin and conducts the power stroke. So in this way, ATP is directly linked to the power stroke. The myosin ATPase hydrolyzes the ATP, and that causes the power stroke. That makes the filaments slide by one another. So that's something really different also. Remember, in skeletal muscle, ATP is not directly involved with the power stroke. It exists to detach the myosin cross-bridge from the actin, and then when it's hydrolyzed, it recocks the myosin cross-bridge, and then the myosin cross-bridge attaches and goes through the power stroke. Thin filaments, thick filaments pull on thin filaments pull on dense bodies and pulls them together in a spiral manner. So here's the operating voltage gated calcium channels on the outside. Calcium comes in, binds the calmodulin. It activates myosin light chain kinase, which activates the myosin ATPase, hydrolyzes ATP, provides power for the power stroke. The myosin attaches to actin and goes to the power stroke. And then the filaments slide by each other in that spiral manner. Then there's a couple other strange conditions with smooth muscle. The myosin light chain phosphatase, I think, detaches the myosin cross-bridge from the actin. Enzymes in calcium channels slow, so long latency time and much slower contraction. So long latency time to contract smooth muscle, and the contraction is so it's much slower. Also, it exhibits less tension, I do believe. Then there's this latch grid mechanism, allows the myosin to be attached to the actin for longer periods of time without using more ATP. So you can have smooth muscle contracted for long periods of time, and it's not using more ATP. So it's very economical, and this is called a latch bridge mechanism. It does exhibit tetanus and very resistant to fatigue. Most ATP made aerobically, but the ATP requirements are small. So these qualities, like I said, allows it to be contracted for a long period of time to maintain smooth muscle tone. So remember, the smooth muscle, all of your body is actually always partially contracted. You can have smooth muscle that's completely relaxed. I mean, skeletal muscle that can be completely relaxed. Even cardiac muscle can be completely relaxed during diastole. But smooth muscle is always partially contracted. In blood vessels, otherwise your blood pressure would be fall to practically nothing, and you'd faint and die. And even your small intestine is always partially contracted. That's why it looks a lot longer in a cadaver, because in a cadaver, all the smooth muscle is obviously relaxed. And the small intestine looks a lot longer than it is in a living person, because remember, there are longitudinal muscles in small intestine, and so it keeps it partially contracted and shorter than it is in death. So that's called smooth muscle tone, and it's really important for maintaining function and blood pressure. Response to stretch. Some ion channels are mechanically gated, and stretch leads to peristalsis in the digestion system. So it's a stretch relaxation response. When smooth muscle is stretched, it reflexively contracts. Right? So brief contraction, resistance to stretch, but then relaxes. This is something that allows the urinary bladder to be stretched and hold urine without expelling it immediately. But once it gets stretched to a certain point, then it's a parasympathetic autonomic reflex into the spinal cord to open up the internal urethral sphincter, and then you get that urge that you have to urinate. There's no length tension relationship So force of contraction is independent to the degree of stretch. Doesn't matter how stretched or not stretched smooth muscle is, it always generates the same amount of tension. And that's not true for smooth. For skeletal, and skeletal has an optimal length to get optimal contraction. Not true for smooth muscle. Okay. Plasticity. Smooth muscle adjusts its tension to the degree of stretch. That like contradicts that, but it doesn't. What it means is there isn't an optimal length for stretch. You know, if you stretch a little bit more, it actually will cause more tension generation. Okay. No Z discs cannot have too much or too little overlap of fibers because there are no sarcomeres. So that's why I guess there's no length tension relationship, because you can't have too little or too much overlap. There's no H zones where there are no myosin cross bridges, so there's myosin cross bridges all through the large filament. So I think that's it for muscle. Yes, that is it for muscle. So we're done. So this will be 203 muscle 3. And so we're done with muscle. And so that means we're done with the video lectures for this semester. Right? So I'll see you later.